how these agrobacterium genes have been modified by a scientist so that we can improve the crops. The first one is known as the co-integrative vector. Now, herein you are seeing two plasmids. This is the disarmed plasmid and this is the intermediate plasmid. If you look at the disarmed plasmid, we have the right border, the left border, okay, this is the border, and the virgins. In the intermediate plasmid, we have the DNA insert, that is our gene of interest, which we want to insert, and then we have the tDNA, that is the transfer DNA. And also, we have antibiotic resistance, that is the neomycin resistance and the kanamycin resistance regions, which is present on the intermediate plasmid. So these are two different plasmid. First, let us see what is the meaning of disarmed plasmid. Now, disarmed plasmid means, as I said in the last class, we know that in the transfer region, there is what is known as auxin and cytokinin in the transfer region of your Ti plasmid in the Agrobacterium. The function of this auxin and cytokinin is to just cause tumor. That is, they undergo replication, they undergo division. The best word to use is division instead of replication. So they form the tumor, and this tumor is called as the crown gall disease. And other than causing tumor, it has no other functions. So what did the scientists do was, they thought that why should that gene be present when it has no function other than just overproducing or overexpressing. So they thought that let us remove this auxin and cytokinin. In this region, let us add our gene of interest. And this is known as your disarmed plasmid. Look at this. This is an example of a disarmed plasmid. Okay. It is named as PGB3850. So, your wild type of Ti plasmid, your wild type of tumor-inducing plasmid, which are present in your agrobacterium, are not suitable as vectors. What are vectors means? They are vehicles. Okay, they are the one who carry the gene from one plant into the another plant. They are not suitable as vectors because of the presence of oncogenes in the tDNA that causes the tumor growth in the recipient plant cell. So this auxin and cytokinin, they are called as oncogenes because they are inducing the tumor in your recipient plant. For efficient plant regeneration, vectors with disarmed DNA are used by making it non-oncogenic by deleting all of its oncogenes. So, if I want to bring about an effective regeneration of the plant, of my recipient plant, without causing any tumor in that plant, it becomes essential for me to remove this oncogenes, that is your auxin and your cytokinin. That should be deleted. The foreign DNA is inserted between the right and the left border and then integrated into the plant genome without causing any tumor. So we remove that oncogene and in the place of that, we add the foreign DNA between the right and the left border. Remember, if there is no right and left border, whatever may be your insertion is, your tDNA, that is your transfer DNA, will get terminated if these two borders are not there. So this is the principle behind a disarmed Ti plasmid. That is, we are removing our oncogene. In place of that oncogene, we are adding our gene of interest. So this is our disarmed. And this is the region of virulence. Okay. So what is the function of the Vir gene is to help in the transfer of your transfer DNA into the recipient plant. Please look at this slide. A disarmed TI plasmid is a type of plasmid that does not carry the gene for tumor induction. An example is PGV 3850, which I showed you now. This disarmed TI plasmid contains the left border and the right border sequencing along with the Vir genes. So what is the function of this Vir gene is? It will catalyze in the transfer of your tDNA, that is the transfer DNA. Where is this transfer DNA present? It is present in the TI plasmid, that is the tumor inducing plasmid. Where is your TI plasmid present? It is present in my Agrobacterium. So this is about the disarmed TI plasmid. The other one what we spoke was intermediate plasmid. This carries the DNA insert, that is the gene of our interest, the desired gene which we want to transfer it into our plant, recipient plant, that is called as a DNA insert, along with the neomycin and kanamycin selectable markers. Now, why I should use a marker? Marker acts as an indicator or to trace the path. For example, if you're going into an unknown wood, okay, a forest, what do? You put a marker somewhere so that you can see that marker and come back to your own path. Similarly, these kanamycin and neomycin acts as a marker which tells us where exactly your DNA of interest has gone, whether it has inserted into the plant material. That is the need for the markers. So it is an identifier or a tracker or a tracer. So along with the neomycin and kanamycin selectable markers, the remaining sequencing on the intermediate plasmid are taken from the tDNA region. Both the plasmids carry some common sequencing which are taken from PBR 322 plasmid from E. coli. That is, we spoke about our disarmed plasmid and our intermediate plasmid. Look at this region. This two region appears to be similar, and this region is a common between your intermediate and your disarmed plasmid. And this region is picked up from your PBR 332 sequence, that is from the E. coli plasmid. So I have a disarmed TI plasmid, and then what did I tell? I have an intermediate plasmid. Now, all this manipulation of constructing your plasmid is done in the laboratory by the scientist. After they have constructed the plasmid, they have to put it back into the bacteria. Only then that plasmid can become active. So the disarmed TI plasmid is maintained and cultured in the Agrobacterium cells. Look at the color, I put the same color. So your disarmed TI plasmid, it is put into the Agrobacterium. Whereas your intermediate plasmid, what is your intermediate plasmid? Actually, your intermediate plasmid is a modified form of E. coli plasmid. Okay, we have taken the E. coli plasmid and we have modified it, and that is called as your intermediate plasmid. And so you put this intermediate plasmid into the E. coli. And afterwards, the E. coli strain carrying the recombinant intermediate vector DNA replicated in E. coli is now transferred into the Agrobacterium. So, you allow your intermediate plasmid to replicate in the E. coli. After you have replicated in the E. coli, then what you do is you take this intermediate plasmid which was there in your E. coli and then you transfer it into your Agrobacterium. So what did I do? I have replicated my intermediate plasmid in E. coli and then I have transferred it into the Agrobacterium. Already my Agrobacterium is containing the disarmed Ti plasmid. Now how will this plasmid or how will this Agrobacterium will look like? This is how it will look like. So this is your disarmed, okay, Ti plasmid, and this is your intermediate plasmid from the E. coli. So the intermediate plasmid, after it has been replicated in E. coli, it is transferred into the Agrobacterium. Now, this bacterium, after receiving your Agrobacterium, after receiving the intermediate plasmid, it is known as the transformed Agrobacterium cells, which carry two plasmid, that is your disarmed plasmid and your intermediate plasmid. The next step is cell selection. What do you mean by cell selection? Cell selection has to be made on a selective media containing kanamycin. The transformed and untransformed agrobacterium cells are incubated in the media containing kanamycin. Only the cells that contain both the plasmid can grow plus the intermediate gene carrying the kanamycin. What it means to tell is after you have done with this procedure, you have to do the cell selection. How I am going to do the cell selection is by using a media, and that media should contain the antibiotic kanamycin. Already we have introduced the kanamycin resistant marker in the intermediate gene. Okay, so this marker is acting as an identifier. It is telling us whether your agrobacterium is transformed or not. So I have done all the manipulation. I'll take all the agrobacterium and grow it in the media containing the kanamycin. So my kanamycin media will allow only those transformed cell of agrobacterium. It will not allow agrobacterium cell that are untransformed. Untransformed means it is not carrying these genes, intermediate genes, or so whichever, if it is not carrying the intermediate gene, it means that it is not being transformed. So only if your agrobacterium is carrying these two, then it gets selected. Okay. So look at this. I have marked it in two different color. That is, one is your disarmed plasmid, another is your intermediate plasmid. Now they are able to grow because why? They are resistant. Even if you have added into the media kanamycin, I can grow on them because I am resistant to these antibiotics, because I am carrying this resistant marker. It is just like you take your polio vaccine. Okay, and when there is an attack from the polio virus, what happens? You don't get the disease because you have been vaccinated. Similarly, what happens here is you already have a resistant kanamycin, so it will grow in this media. So now you see, you somewhere that yellow color is not there. Okay, only the blue genes are, or the blue plasmids are present. That means it indicates that this particular agrobacterium is carrying only the intermediate plasmid because it is also resistant to kanamycin marker. So this is how we select. So your main aim is to select the transformed gene by using the antibiotic kanamycin. Now, when I started with my first slide, what did I say? I told you that we are going to speak about co-integrative vectors. What do you mean by co-integrative vectors? Co-integrative vectors means there is an homologous recombination. Okay, there is a homologous recombination of the bacterial plasmid with the tDNA region of an endogenous Ti plasmid in the Agrobacterium. What we mean to tell is there should be an homologous recombination between your intermediate vector and your disarmed vector. And what is this homologous region? Your homologous region is this PBR 322, which was derived from the E. coli. See, these both are same. Okay. See, in this first picture also we told that we have introduced this common sequence. Now you know the importance of the common sequence. Why I introduce this common sequence means I can integrate these two because these both are having the same sequence, they can get integrated. Finally, I get a co-integrative vector. Look at this homologous recombination between PBR332 sequence. So this is my sequence. This is my transformed T plasmid. So now what I do after I get the co-integrative vector, I will insert this again into the agrobacterium. Okay, I have integrated into the agrobacterium. Next, what should I do? Come here. So this bacteria, this is your agrobacterium containing your co-integrative vector. Now we are not going to use disarmed or intermediate. We are going to use one term known as co-integrated vector or your co-integrated plasmid. After this has been done, what the scientists do is they take a media and into the media they add the agrobacterium containing your co-integrated plasmid. That is, it should have your PBR 322. It should have your DNA of interest. It should have the two markers, that is your neomycin and kanamycin. The Vir gene, which is present on your co-integrative vector, that also should be present. And then along with this, you're adding your plant cells. Which are these plant cells? These are the plant cells which are of interest. Suppose say maybe this is a crop plant and I want to modify this plant with my gene of interest, which is there now in the agrobacterium. So I will have a mixture of this, having the mixture of agrobacterium and my plant of interest. Next, what happens is you make a slight wound in your plant. Now, what happens? Your agrobacterium will get attracted to the signal from the whatever the phenolics which is released from your plant. So it will enter into your plant through the Vir genes. We'll not talk about the genes. We have spoken it enough in the previous class. Okay. Now, there are two probabilities. Your plant cell may be transformed. That means it may get integrated with this agrobacterium co-integrative vector, or there are chances that it may not get transformed. Now, how I'm going to identify which is being transformed and which is not being transformed with the co-integrative vector? For this, I have another media. This is also known as selective media. In this selective media, what I'm adding is I'm adding my neomycin. So the plant cells that are transformed and untransformed are incorporated into the neomycin media. Only transformed plant cell with the DNA insert and the selectable markers will be able to grow in this media. That is, only this can grow. Okay? That is, only your transformed or genetically modified plant can grow, and those cells which are not being transferred, they cannot grow. This is what I have shown you. This is an untransformed plant. Integration has not happened, whereas this is a transformed plant. See, it is a neomycin resistant gene it is carrying. It is carrying your transformed plant vir genes. It is carrying your borders, your PBR 322, your DNA of interest, and the two markers. So, this is about your co-integrated vectors. So here you started with two vectors, intermediate vector and disarmed vector. And then what you have done is you fused them. That is, there is a homologous recombination region. Using this recombination region, you fuse the both the plasmid to get a single transformed plasmid, which again you insert it into your agrobacterium, and then you try to insert it into a plant of interest. And using the markers, we can select